Hi, everyone. It is really a great honor for me to present our studies in developing drug for pancreatic cancer. So by way of introduction, I am a medicinal chemist. We are interested in developing small molecule drugs for the treatment of various diseases, specifically developing a drug for pancreatic cancer, as well as glioblastoma and ovarian cancer. And today I will be talking mostly about our recent effort in designing drugs for pancreatic cancer. So we are interested really in the development of first-in-class inhibitors against select targets that we have been playing for the past few years. And if you're interested, we can talk about other targets in other time. But today, I would like to focus on a very exciting research area that on a very exciting class of compounds that we discovered from a phenotypic screen against pancreatic cancer. So I'm not going to go too much in detail explaining the disease itself, but I just want to remind you, so pancreatic cancer is kind of different from other cancer. It's very desmoplastic and is very hypoxic in a very hypoxic environment. And this hypoxia is actually generate lots of cancerous stem cell. And if you look at the microenvironment of the tumor, about 10% of the cells are really pancreatic cancer cells. The 90% of really what we call the stroma and all the connective tissues, all the immune cells. But the disease is really a KRAS disease. So pancreatic cancer is, over 90% of the patient, they have mutation in KRAS. So pancreatic cancer to me is a KRAS. So one of the function of KRAS is really to upregulate glucose metabolism. So glucose it is doing that by increasing the levels of glucose transporter, in this case GLUT1, but it's also GLUT3 as well. But as a result, really, essentially all the enzymes involved in glycolysis are also upregulated. So as you know, remember from glycolysis, the purpose is to generate pyruvate, which goes in a TCA cycle to generate ATP. Now, the other side of glycolysis is really what we call oxidative phosphorylation or OXPHOS. So glycolysis process itself, it only generates 2 ATP, whereas the ETC electron transport chain or OXPHOS, it generates 32 ATP. So that's the reason we say mitochondria is really the powerhouse of the cell, because it can generate lots of ATP. So if you could imagine that, so these are the major pathway for metabolism in the cell. And so during the past 40 years, people have been trying to target glycolysis. And essentially every single enzyme in the glycolysis pathway has been targeted by different drugs. So far, after 40 years of research, we really do not have a drug that is working in the glycolysis pathway. Every single one of these drugs have failed for lots of different reasons, which I do not have time to go into. However, now the field is changing toward developing drugs for OXPHOS, for targeting the electron transport chain. The OXPHOS machinery is really very, it's just a beautiful machinery, which is made of several different complexes. And this is a very well-established process that composed of 5 complexes, complex 1 to 5. Essentially, what it really does, it converts NAD to ATP at the end of the cycle. Essentially, it's transferring electron from complex 1 to 2 to 3 to 4 to 5. So each one of these complexes are made from different genes, and as you can see, complex 1 is quite sophisticated. Is made of quite a few genes. So during the past several years, we have had success in producing compound that can inhibit different complexes, but those are all were lab reagents. They were not really drugs. But now we have small molecule drugs that are really selective for one of these targets. So that is what I will be talking about. So during the past several years, there are several very high-profile papers were published that showing, for example, in this case, the oncogene ablation-resistant pancreatic cancer, they become very sensitive to OXPHOS, and lung cancer cells with mutation in SNF complex, they are extremely sensitive to oxidative phosphorylation inhibitors, and also in a CML that the so-called residual disease, when they become a cancer stem cell, they can be quite effective to eradicate them by inhibition of OXPHOS, and also in a mantle cell lymphoma, OXPHOS is very important. And also in P10 null cells, so those cancers with the P10 null, they become extremely sensitive to complex 1 inhibition. And pancreatic cancer stem cells are particularly sensitive, as well as prostate cancer cells that when they remodel in high-grade prostate cancer. And so as a result, this mitochondrial metabolism is becoming a really very exciting area to drug. And so how and where do you use oxidative phosphorylation inhibition? So we know for a fact that select cancers, they are quite sensitive to OXPHOS inhibitors. And we have a collection of biomarkers that we can select patients to give them OXPHOS inhibitors. And cancer stem cells are really dependent on OXPHOS. And also another way to restore anti-cancer immunity is by blocking or inhibiting OXPHOS in immunosuppressive cells. And drug-resistant cells, specifically cells that they become resistant to kinase inhibitors, they are becoming dependent on OXPHOS. And because of this, we are using a synthetic lethal approach to develop inhibitors so that we can combine with OXPHOS inhibitors. So I will show you only one example of a drug right now in phase 1 clinical trial for MD Anderson. If you can see here that the cells that are wild type in SNF, they do not respond. Whereas cells that are mutant, every single one of them, they respond beautifully to this drug. And this is not only in the cells, but also in the PDX models with the SNF mutant, they respond very nicely. And also in cells that are glycolysis deficient, in this case, neuroblastoma cells, they respond very nicely. And the cells that are resistant to ibrutinib, they respond very well to this drug. So IACS compound drug right now is in phase 1 clinical trial, and early studies is showing that it's well tolerated, and preliminary evidence of anti-tumor activity is seen in leukemia. Now MD Anderson trying it in the different solid tumors as well. So with this background, we started the high throughput screening of a collections of our compound. We have 40,000 compounds in my lab that are highly diverse, that they represent 10 million compounds. We tested 4,000 of these molecules in galactose versus glucose enriched media. We identified this compound called N18230 as a lead molecule. So this particular compound is quite potent in the galactose media versus glucose, and this structure is shown here. So this molecule has a chiral center. We synthesize both the R isomer as well as S isomer. And we showed that the R isomer is remarkably more potent than the S isomer here in inhibiting cell growth. So whenever you see this kind of differences just by only one differences really, so that you have an isomer that is essentially this group is above the plane and this group is below the plane. So we're really dealing with a very specific pocket on the target that is this particular molecule is binding. So we were very excited with this compound DX2201 that we tested this compound in 90 different cell lines. Here showing you NCI60 result, and some lung cancer cells, they respond. Leukemia cells, many of them, they respond. Like one colon cancer respond. The CNS cancer, they do not respond a whole lot. Pancreatic cancer, prostate cancer, they do not respond. As you can see, there are select cell lines that respond remarkably well to this drug. And we also tested to additional 30 cell line in my lab, and we see that testing both in the normal cell as well as leukemia cells. So unfortunately, NCI60 does not have a pancreatic cancer cell line. So we tested several pancreatic cancer cell line in my lab, and most of them, they respond. And some of the ovarian cancer cells respond as well. So we got very excited with this result, and we tried to see what happened at the RNA level. So we used this bromouridine sequencing. It's just essentially RNA-seq looking at the synthesis of nascent RNA. And as we expected, we see the top gene set that is affected is oxidative phosphorylation, that essentially every single gene in an OXPHOS panel, they go down and glycolysis goes up. We also did proteomics, and when we overlap proteomics data with RNA-seq data, we see very exciting common gene set that they go down or they go up. Specifically, I would like to bring your attention to this sets of genes that are implicated in DNA repair and pyrimidine and purine biosynthesis. So all of these gene sets, they go down, and I will explain later on what is the significance of this. So to see exactly where the drug really goes, we developed, we synthesized a fluorescent labeled analog of this compound. And then we can clearly show that it's localized in mitochondria, as we expected. And to understand the mechanism and also to identify the target, we generated 6 different resistant clone, and you can see this cell lines. Now the new resistant cell lines, they're highly resistant to the drug in the presence of glucose as well as galactose media. And what is really interesting to us, the IACS compound, the MD Anderson drug, is equally potent in resistant cells as well as sensitive cells. So what it means that our compound has a completely different target than the IACS compound. I mean, we kind of expected that because structurally they are very different. And also, they are very different mechanism from other complex 1 inhibitors such as metformin and fenformin and rotenone and antimycin. So we are dealing with a class of compound that are really unique, has a novel target, and structurally are also unique. So we can show clearly that it shows inhibition of complex 1 in nanomolar range and is not active in the resistant clones. And when we expanded our studies to look at the cell lines that we have in the lab from patient-derived pancreatic cancer cell lines, so these are all pancreatic cancer patient-derived cell line that we have in the lab. As you can see, some cells, they respond beautifully, some are more resistant. And same thing with the cell lines that are commercially available. And what I would like to show you here, this HPDE cells, so these are the normal pancreatic ductal epithelial cells, they do not respond. So it's not really killing all the normal cells. And we show that it decreases the mitochondrial mass and it produces a reactive oxygen species, and it kills the cell through apoptosis. So as I mentioned earlier, that tumor microenvironment of pancreatic cancer is really hypoxic. So we developed a 3D spheroid assay that you can really see that spheroid cells, and when we use the hypoxia probe, you can see in the 3D spheroid that the cells are quite hypoxic. You can see very nicely. But when you add the 0.4 micromolar of our drug, this hypoxia is really is gone. So it's no longer hypoxia. So this is exactly what we really want for a drug to treat pancreatic cancer because of this hypoxic microenvironment. What is also another added benefit to the drug is actually decreasing PD-L1 expression. So as you could imagine, so this is what you really want to do. You want to combine this potentially with immune checkpoint inhibitors. So that's a direction that you're moving forward. And in fact, it is already known that metformin, which is a complex 1 inhibitor, is inhibiting electron transport change in MDSCs as well as macrophages and T regulatory cells. And so we would like to try our compound as well in a syngeneic model of pancreatic cancer cell lines. So we tried to get as many mouse cell line that we could find. Unfortunately, there are not too many mouse cell line to test our drug, but the one that we tested, we show that we see that these PANO2 cells are extremely sensitive to the drug. And as you can see, compared to Miopaca, for example, that we were using earlier, the PANO2 cells are very sensitive to the drug in a colony formation assay. So because of this, we'd see, okay, what happens in the mice? So you can see that clearly the compound showed beautiful in vivo efficacy. It's as comparable as a IACS MD Anderson compound with the same concentration. And we do not really see any sign of toxicity. What is really exciting with our compound is highly synergistic with the IACS drug, as well as synergistic with 2-deoxyglucose and with an inhibitor of glycolysis pathway. And we tried a larger panel of FDA-approved drugs, as well as drugs under clinical trial. We can see quite a few of them are synergistic, and several of them are additive. So I mentioned earlier that DNA repair gene set and pyrimidine purine biosynthesis gene set, they all go down in both BRUSIC as well as proteomics. So this is really a perfect place to really combine such drug with radiation. And when we do that, we can see a very nice synergy with radiation, and that's kind of expected. So potentially we could combine these OXPHOS inhibitors with radiation. So that was our original compound that we identified from the screen. Obviously, to make it into a drug, we need to do chemistry. So this is the structure. Unfortunately, it is metabolically not stable. It's only the half-life in a mouse liver macrosome or human liver macrosome is only a minute. As a medicinal chemist, you recognize this as an ester group, so immediately we change it to amide, and we can easily increase the macrosomal stability to 27 minutes, and then some other analogs to 41 minutes. So we tried to make a large number of analogs to understand, to optimize physicochemical properties of these molecules, with the idea that we want a compound that is less than 100 nanomolar. And as well as stability, the half-life is over 30 minutes. So we have made a large library of compounds. We also identified the target that we are preparing a manuscript right now. I'm not going to tell you about the target itself, but I can tell you we know exactly which part of the molecule bind to its target, how all the hydrogen bond interactions. So using a structure-based drug design, we have designed 200 analogs trying to make compounds with a better metabolic profile and also higher potency. That's what I will be showing in the next few slides. So we have generated lots of different derivatives, essentially making a substitution on every single part of the molecule. And as a result, we have compounds that are metabolically more stable, like for example here over 60 minutes, and these two over 60 minutes in both mouse and liver macrosome. So these molecules are potentially very potent, and so we're talking about really 11 nanomolar now as this example, and is quite stable. So in conclusion, we have first-in-class inhibitors of this target that I would like to discuss in the future, hopefully. And we generated lots of analogs that will show that it's synergistic with select drugs as well as radiation, and extensive MedChem campaign, established SAR. So we know exactly how these molecules work, how they bind, where to improve, and over 200 analogs. Some of them are quite potent, and metabolic stability is favorable. And so one of the most potent compounds is that it's 9 macromolar. So this is one of the most potent OXPHOS inhibitor reported in the literature. And we have analogs that are quite synergistic with mice in a different model, and all of them are protected by a patent application. So what do we have so far? So we have this class of compounds that are active in 90 cell lines. We have done a very extensive lead optimization and genomic and proteomics mechanism of action studies, in vivo proof of concept studies. So we have done all of that, and obviously I cannot show you all the data, but if you're interested, please let me know. So what we have in hand right now, we have at least one compound that we call it is a clinical candidate that essentially is ready to go. And we have 4 other backup compounds that just in case something happened, it's always nice to have a backup compound. And we are preparing several publications in this area. So we are really hoping to partner with pharma to take it to the next level, because the expertise of my lab is really here. And the rest, we really do not have much expertise in academia. So I would like to thank the people who really did all the work. Ding and Ibin were really the work on this for 5 years. And then also joined with Armita and Joita, who really contributed significantly to this area. And that's where I stop. Thank you very much.